Today we will be talking about Chapter 6, which is all about cell membranes, and have this lecture divided into two parts. So in part 1, I am basically just going to focus on the structure of cell membranes, talk about some of the various parts that are found in membranes, different components, and then in the second half we are going to focus on how things get across membranes. And keep in mind, because every cell is surrounded by a cell membrane, um, the way things cross membranes is really the way things get in and out of cells, which is key for keeping cells alive and allowing them to function properly. Okay, so here we have a little cartoon drawing of a cell membrane, and you'll see membranes referred to as fluid mosaics. And so what that means, if obviously the term fluid is pretty self-explanatory, but you can see some of the various components of the membrane here, the phospholipid bilayer. These brown structures are proteins that are associated with the membrane. We have cholesterol molecules in yellow here, and then we've got some various components of the extracellular matrix on the outside of the cell, and maybe some cytoskeletal components inside the cell here. But the fluid part is referring to the fact that all of these components are constantly moving, right? So the phospholipids can change places with each other, so a single phospholipid could move around within the membrane. Even these proteins or cholesterol molecules that you see here are basically like floating around within the plane of the membrane. So it is very dynamic, very fluid. And the mosaic part of that term, of the fluid mosaic model, is just referring to the fact that you do have lots of different parts, right? A mosaic, you might think of like a tile mosaic, where you've taken little pieces of colored tile and arranged them to create some other image. So here, your pieces are the phospholipids and the proteins and the cholesterol molecules, etc. So we have a fluid mosaic made up of these basic components. And just to remind you, I know we've already looked at phospholipid structure, but they have this unique amphipathic structure, meaning you have what's considered the head domain up here, which is very hydrophilic, likes to interact with water molecules because of these charges that are found on the choline and the phosphate group. And then you have the glycerol fatty acid tail portion, which is very hydrophobic because it's basically just hydrogens and carbons for the most part. There's no positive or negative charges to be found, so it does not like to interact with those partial charges that you find on water molecules. So again, once you put those amphipathic phospholipids into an aqueous environment, they will spontaneously form this bilayer with the hydrophilic heads happy to interact with the water, and the hydrophobic fatty acid tails facing each other, basically trying to get away from the water molecules. So we have that as our backbone, and then on top of that, we're going to lay our proteins and our cholesterol molecules, and maybe some sugars or other things that might also be associated. So one thing I wanted to point out, just while we're talking about the lipid part, the phospholipid part of the membranes here, is that lipid composition can vary from organism to organism, or even from cell to cell within one single organism. So how could that happen? Well, the phospholipids that we've looked at so far, we've really just looked at this one cartoon. They always look the same, and they are always going to have the same general structure. But there's really a couple of ways that phospholipids could vary one to another, and it all has to do with what's going on with the hydrocarbon tails or chains down here. So those chains could be longer or shorter. They might have more carbons or less carbons, and they could be saturated or unsaturated. So if you remember that discussion a couple of chapters ago, if you have any double bonds in this chain, carbon-to-carbon double bonds, then it would be an unsaturated fatty acid with a bend in the chain. and that's going to prevent the phospholipids from packing together quite as tightly. So depending on what those fatty acid tails look like on the phospholipids, you could actually, vary the characteristics of your membrane from one cell to another quite a bit. And then another thing to keep in mind is the amount of cholesterol that's found in a membrane can be quite variable. So let me go back to the cartoon showing you those cholesterol molecules here. So what cholesterol is doing in the membrane is it's almost like a buffer of sorts, helping to maintain kind of a constant level of fluidity, right? We talked about this being a fluid mosaic model. And the membrane's sort of walking a fine line, because if it gets too fluid, it just dissolves away into nothing, right? Basically comes apart. And if it becomes too thick, then the membrane just can't function properly. So it needs to maintain sort of a consistent level of fluidity, if that makes any sense. And the cholesterol is actually helping with that process. So if you have a membrane that's more fluid, the cholesterol, which is kind of wedged in between the phospholipids, is sort of acting as a binding agent, kind of helping to hold things together. So it prevents it from becoming too fluid. And on the flip side of that coin, it can also prevent the lipid bilayer from packing together too tightly, right? So these cholesterols in between the phospholipids will stop them from being pressed together too closely, and the membrane becoming too dense or too thick. Okay, so it's really helping to mediate the level of fluidity, and again, that is key for the function of the membrane. So some membranes, if they're in a, maybe exposed to high heat, which would increase fluidity, they may have higher cholesterol content. It could be up to 25% cholesterol in some documented cases. All right, so I think we've already talked about, we've got this idea of fluidity under our belt. I did just want to point out that the fluidity can be affected by some environmental conditions like temperature as well. So the warmer it is, the more fluid a membrane would tend to be. The colder it is, the more dense it would tend to be. Okay, so we've got our lipids as that base backbone of the membrane. And now we've got, as we saw in our cartoon, quite a few proteins that may be associated with any given membrane. And there's really two categories of membrane proteins. There's what are called peripheral membrane proteins, peripheral meaning on the edge. So these would be proteins that are just associated with either the exterior or the interior surface of the membrane, but they don't actually penetrate into the hydrophobic center of the membrane. Whereas integral membrane proteins do cross into the center, and in fact some integral membrane proteins may extend all the way through the membrane and would be considered then transmembrane proteins. Okay, so peripheral versus integral. So actually, before we talk about this, let me go back to that other picture real quick. So like this would be an example of a peripheral membrane protein, at least looks like it from the image here, where most of this protein is just associated with one side of the membrane. Whereas these that we see up here, these are all integral membrane proteins, because we have at least part of the protein here inside the hydrophobic interior of the phospholipid bilayer. Let's go back up here. So here's our transmembrane protein, right? And if we were to actually look at the amino acids in this polypeptide chain that make up this protein, you would see that all the amino acids here in this alpha helix that are in the middle of the membrane are all going to be very hydrophobic amino acids, and they would need to be able to stably hang out in this hydrophobic interior of the membrane. And then the amino acids that are out, I guess, in here inside the cell or out here are going to be very hydrophilic amino acids probably have some charges of some sort. So again, that one we were just looking at, that protein would be an example of an integral membrane protein that is also considered transmembrane because it extended all the way through the phospholipid bilayer. And just to remind you, right, all these membrane-associated proteins, even the transmembrane proteins, can move about freely within that bilayer. So, there are some examples of proteins that do get anchored into specific regions, but in general, they can move very freely in that fluid membrane. And I wanted to show you just quickly a cool and simple experiment that was used to initially demonstrate that membranes are fluid. And what researchers did was in the lab, they fused a mouse cell with a human cell, and were looking specifically at the transmembrane proteins that were visible on the cell surface. And so, of course, the mouse cell has mouse proteins. They had a way of distinguishing mouse from human. Human cell has human proteins. And initially, when you fuse those two cells together, and cells can be induced to fuse quite easily because those membranes will quite happily mesh together, you end up with this cell where right after the fusion event, right, basically half the proteins on the surface are mouse proteins, and then on the other half are the human proteins. And so what researchers found is very quickly, in a matter of minutes, they would see that the mouse and the human proteins, membrane proteins, were evenly distributed across the whole surface of the cell. And so the only mechanism that could explain that is if those proteins are moving around very freely and in fact quite rapidly in the membrane. Because really the only other possibility would be, oh, well, the cell just starts generating new proteins and sending them to the cell surface, but it would basically take too long for that to be a reasonable explanation. So the fact that the membrane proteins move around so quickly really demonstrated, yeah, membranes are fluid, these proteins can move. And that shouldn't be surprising to you given what we've already discussed regarding membranes inside of cells. So you might remember the endomembrane system that we talked about. And I was trying to get across the fact that you could have vesicles that would basically bud off of the endoplasmic reticulum, and then those membrane-bound vesicles could fuse with the membrane of the Golgi, and vesicles could bud off the Golgi and fuse with the plasma membrane. So this idea that membranes are constantly changing and capable of fusing with each other, very dynamic structures, is one of these hallmarks of biological membranes. Okay, so we've got our phospholipids as the base of our membrane. We've got these proteins associated, either peripheral or integral membrane proteins. And then we also will have a fair amount of carbohydrates, sugars, primarily on the exterior surface of the membrane. So these can be directly attached to the phospholipid heads. In this case, those phospholipids would now be considered glycolipids, that glyco prefix just meaning carbohydrate. But a lot of these sugars are actually going to be attached to some of those membrane-associated proteins, making them glycoproteins, so sugary proteins. And that's going to segue into the extracellular matrix, right, which has a lot of carbohydrates associated with it as well. In animal cells or in plant cells, you have the cell wall outside the plasma membrane that's basically almost entirely cellulose carbohydrate. All right, so the last thing I wanted to talk about here in terms of structure of membranes are some of these specialized junctions that we find in cell membranes that can be used to anchor cells together, right? And so this is critical in multicellular organisms where we need to hold cells together and organize them into tissues. Um, and we have these three types of cell-to-cell junctions that I want you to know about, what are called tight junctions, desmosomes, and gap junctions. And I'll give you just a quick description of each of these. So you can think of tight junctions as basically quilting um, between two membranes, or stitching that would be holding those two cells, those two membranes together. So what you're looking at in this little image here are the membranes of two different cells pushed up against each other, right? So we'd have the space between the two cells, kind of right through the middle here. Um, and the little purple dots here are the tight junction, and it's kind of this linear protein stitching that goes through both plasma membranes and is anchoring them together. Okay. So there's sort of two effects of tight junction. One, as I just said, is it will hold those two cells together because they are literally kind of sewn together with these proteins. Um, and then the other effect is it can help control the movement of fluid or molecules that are outside the cells, like moving between the membranes of the two cells, right? So you imagine you've got your cells surrounded by extracellular matrix and fluid, and there's all kinds of molecules that may be moving around outside the cells. So imagine in this instance you have some molecule that's trying to move from top of the image to bottom, like just between the two cells, between the two plasma membranes. So what's going to happen as it flows downward, um, eventually it's going to bump into this region where the tight junction is holding those two plasma membranes together, and now it just can't move down anymore, right? It's blocked, and it's going to force that molecule or the fluid to move now left or right. Okay, so tight junctions are often used in tissues and in organs to kind of direct the flow of molecules and fluid between cells. Then the second type of cell-to-cell junction are the desmosomes, and you can think of these as basically like spot welds, essentially. Um, and you have this big protein weld, right? That's anchoring the two plasma membranes together, and you see all these green, looks like green hairs coming out. So these are keratin fibers, and you might remember intermediate filaments, right, are the type of cytoskeletal fiber that's made out of keratin. So these are really just intermediate filaments that kind of flow into the desmosome. So these desmosomes are really just exclusively used to fuse two cells to each other. We're not going to limit the movement of any molecules outside the cell between the plasma membranes, because if we go back to the same example we were just using with the tight junctions, if some molecule is trying to flow from top to bottom in this image, it can easily do so, right? Even if it bumps into this desmosome, it can then just flow right around it, right? So it's a little less restrictive, but just another possible mechanism for anchoring these two cells together here. And then the last type to mention are what are called the gap junctions. And gap junctions, they do have the effect of holding two cells together, but their main purpose seems to be allowing communication between two adjacent cells. And you can see in the picture here, the gap junction is really a little pore um, that will span the plasma membranes of both of these two cells. And so you could have small molecules in the cytoplasm of the cell on the left here that can now move right through this gap junction into the cell on the right. And you might have a whole chain of cells that are all connected with these gap junctions, and we see them used in tissues and situations where we need cells to be able to sync up and communicate with each other very quickly, right? So they could do that by passing signaling molecules um, through those gap junctions in a very quick manner. Okay, so three types. Tight junctions are the quilting that can help control the flow of material between two cells on the outside of the cells. We have the desmosome spot welds that are really just being used to anchor the two cells together. And then we have the gap junctions that are going to allow small molecules to move from the cytoplasm of one to another, one cell to another. Oh yeah, I forgot. So this is really the last thing I wanted to mention in this chapter, is talk just a little bit about what's going on at the cell surface in terms of interaction between the plasma membrane components and the extracellular matrix. And I wanted to point out this one key protein that's called integrin. And so this would be an example of a transmembrane, integral membrane protein, right? You can see it spans all the way through the membrane of the cell. And on the inside of the cell, part of this integrin protein is connected to the cytoskeleton, in this case microfilaments. And then on the outside of the cell, part of the integrin protein can connect with fibers of the extracellular matrix. But interestingly, the integrin proteins can change their shape, so they can sort of be folded up like this, where they're really kind of inactive and they're not interacting with the extracellular matrix. Or they can, stand up, kind of raise their head up in the air and grab hold of those extracellular matrix fibers. And it turns out this is really important for cell movement. If you remember in a previous chapter, we talked about the fact that for a cell to move, which happens all the time in multicellular organisms, important during embryonic development, cancer cells move very readily, so it's something that biologists and medical researchers are really interested in understanding. We talked about what was kind of going on inside the cell in terms of this interaction of microfilaments and myosin motor proteins kind of squeezing the back end of the cell and pushing the plasma membrane forward. So just to add one more layer to that story, now we have the integrin proteins on the cell surface, and you can think of those as kind of like the treads on the cell's shoe almost, that are giving it traction as it tries to move through the extracellular matrix. And this also ties in with our discussions of the membrane being very fluid and the fact that these proteins can move in the plane of the membrane. So this is what happens with the integrins and how they help drive cell movement. Is you will have integrin proteins that get deposited in these vesicles at the very front, the leading edge of the cell. So this cell is moving off to the right here, right? So this vesicle fuses with the plasma membrane up here, and now all of a sudden these integrin proteins are on the front surface of that cell. They change their conformation so that they can grab hold of the extracellular matrix, right? So giving the cell that traction. And now because the membrane is fluid and the cell is kind of pushing itself off to the right, that integrin just kind of flows through the membrane gradually back towards the back end of the cell, where it then kind of gets internalized and sent back up to the front again. So it's almost like tank treads, I guess would be the best analogy, just kind of rolling around and providing that interaction, that traction with the extracellular matrix. All right, I will stop there and I will pick this up in part 2, where we will talk about how things get across membranes.